Bismillah ar-Rahman ar-Rahim. Ahlan bikum. This is our last lecture, and it is going to be mainly about two groups of multicellular parasites, and these are the cystodes, known as tapeworms, and the trematodes, which are called flukes. From the name tapeworm, it means the worm is comparatively long, and it is thin in structure. So these parasites could be up to 10 or 15 meters long, depending on the species, and the body is made of many units known as segments or proglottids, and all adult trematodes are found in the intestine of vertebrates, while the larval stages could be found in many invertebrates as well as in the vertebrates. In general, the body is flat, white or somewhat yellowish, and is divided into units called segments or proglottids. That characterized all important tapeworm infecting man are characterized by a head which is called the scolex, and the head is usually provided with suckers, and it may be with hooks or both suckers and hooks. Another species, for example, belonging to another group known as pseudophilidian, because all the important ones in the order Cyclophilidia, which is characterized by the scolex, which is somewhat rounded and having four suckers, and in the middle there may be a retractable portion known as rostellum, and this rostellum may be provided or armed with hooks and spines. In the pseudophilidian, we have one important fish parasite, and instead of having suckers, it has just undulations or grooves, two grooves, one on the ventral side and the other one on the dorsal side. And the function of the suckers, the hooks or grooves, is for attachment to the intestinal surface. This diagram to the left shows you the scolex of Canis saginata, the non-beef tapeworm, which is infecting cattle, and beside it is the scolex of Tenosolium, which is the pork tapeworm infecting pigs and other rodents. And here you could see in the scolex of Tenosolium, there is almost a row of spines or hooks. The body starts with the scolex, which could be provided with, as we said, four suckers or suckers, rostellum, and hooks, and then it is followed by a region where the segmentation is not clear. This we call it the region or the region of growth or the region of proliferation. So usually you don't see segmentation, and usually it is the newest part of the worm. As you go down the worm, you will identify a region with a couple of segments where you don't see male or female reproductive system, and we call it a region of young segments. In the middle, you will find almost a region containing many segments, and they're showing both male and female reproductive system. As you can see to your right here, there is two lobes. These are lobes of the uterus, and the other structures are male and female structures. The last part of the worm, which is the oldest part containing the oldest segments, usually the female uterus elaborates, and the other male and female reproductive system, they disintegrate. So the old segments, they just become sacs full of eggs. The uterus will be highly branched, and it shows many branches, and all of them are just filled up with eggs. If you look to the egg, it is almost here to the upper right in gray color. It is rounded and with thick eggshell, and there is many striations. So it is characterized by being rounded with comparatively thick striated eggshell. This stage, for example, if you look to the last, the gravid proglottids, or what we call RIPE or IPE proglottids, on both sides, the one to your left with so many numerous branches is Athena saginata RIPE or gravid proglottid, and the one which is just above the word gravid proglottid is comparatively having less uterine branches. There may be about 15, while it could be up to 25 or 30 branches, lateral branches in case of Tinea saginata. So usually just the number of the uterine branches, that's the branches of the uterus, can help you to identify which is which. Secondly, of course, identification is based on recovery of eggs, or of course, one or two or a group of segments which could be passed out in the feces or in the stool of the patient. However, it is not easy to say whether this egg is a Tinea saginata or Tinea solium or another group of parasite known as a Echinococcus species, because all of them are rounded and having thick striated eggshell. So it may be epidemiologically, on this area or this community, possibly the only parasite or the only tapeworm is Tinea saginata or Tinea solium. What about the life cycle of these two parasites, Tinea saginata, the beef tapeworm, and Tinea solium, the pork tapeworm? Usual infection is by ingestion of an embryonated egg where there is a small larva inside the egg. So when the eggs are ingested by a cattle in case of Tinea saginata, or by pig in case of Tinea solium, the small embryos, the eggshell will break in the intestine, and the small embryos will be carried by blood to different organs. So usually it leaves the intestine of the animal and it goes to different organs, and they develop into what is known as bladder worms. Technically, they are known as cysticerci. Each one is a cysticercus. The bladder worms are usually just fluid-filled vesicles containing scolex. So the bladder worm is a fluid-filled vesicle containing an invaginated scolex. So when man eats undercooked beef or pork containing those bladder worms, then the meat will be digested and the bladder worm will be released. So the scolex will be coming out and it's attached to the intestine. It loses the bag and it starts to feed to be another parasite. Okay, so in the intestine of man, the ingested meat will be digested and the cysticercus or bladder worm head will evaginate and it attaches to the intestinal mucosa and starts to feed, and it could be a worm of a couple of meters. Tinea solium, if they are ingested by man, the eggs will release the small embryo in the intestine, and instead of staying in the intestine, they will move out to 4 bladder worms in the human body, anywhere. But usually in this case, they cause bladder worms because they enter circulation and possibly the hepatic portal vein, so they will be carried to the liver, and they could be forming bladder worms or cysticerci in the liver, cysticerci in the heart muscle, or even the brain or lungs. So usually infection by a larvae or by larvae of tapeworms is more pathogenic because they are causing infection possibly in sensitive areas, tissues, or systems, unlike the other parasite which is found in the lumen of the intestine. So precisely, ingestion of Tinea solium eggs by many or by man, they develop into bladder worms in various muscles. But ingestion of Tinea saginata by man, the eggs will be just digested or passed out. So I just wanted to say precisely, Tinea solium could cause infection when we ingest the egg and they just become larvae as they develop in the pig, or they can cause adult worm infection when we eat pork containing bladder worms. So the pathology of beef and pork tapeworms, adult cysticercosis is usually asymptomatic. That is infection by an adult parasite, which is usually living in the intestine. It could be asymptomatic. It depends, for example, how big or how small the worm and how many worms are there. But still, it may cause abdominal cramps and pain. However, the infection by the larvae, that's what we call Taenia solium infection, when ingesting the eggs of Taenia solium, is more pathogenic because it creates, for example, space-occupying lesions in the brain, in the liver, in the lung, etc. So in the brain, they can cause headache, and they can lead to vomiting, eye problem, or even retinitis. If you see to your right, we have cerebral cysticercosis. That is, the cysticerci of Taenia solium are developing in the brain. And it is the most common epilepsy-causing disease, infection of brain with Taenia solium cysticerci. Diagnosis of adult worm infection is usually by seeing the eggs or proglotid in the stool of the patient. And treatment, traditionally there is what is known as nucleosamide. However, recently praziquantel, which has been developed as the treatment of all flukes, is also found to be effective against tapeworm infection. So precisely for treatment, traditionally nucleosamide is a drug of choice, but now nucleosamide and praziquantel both could be used for, each one could be used for treatment. How to prevent? Proper disposal of human excreta, because animals, cattle, and pigs are infected when the eggs are passed in the stool of infected patient. Adequate cooking of beef and pork. If you cook the meat well, or the beef and pork well, then you kill the bladder worms in the meat. As I said, there is a parasite which is also causing infection of man. It is usually found, the larval stage is found in fish, and this is known as Bifidobothrym latam. So freezing also of meat, as well as freezing of fish at -10 degrees for a day or more, it can kill the bladder worms in meat, that is in the beef, in the pork, as well as in the fish. This is a group of what is known as flukes. I just wanted to say we have different types of flukes, which are trematodes that can cause infection in the blood, like the schistosomes, the one to the upper left, which looks somewhat rounded. In actuality, the male is somewhat flat, but it is curved, forming a groove where it carries the slender, thin female, as you see in the, as it is labeled. Then you have to your right, Fasciola hepatica, and this is a parasite in the liver, or usually it lives in the liver. Down below it is Paragonimus westermani. The other parasite is somewhat globular, and it usually is found in the lung. It is known as the Chinese lung fluke. Down is the smallest trematode infecting man or fluke infecting man. It is known as Heterophis, and it is usually found in the intestine. And Fasciolopsis busci is the largest trematode infecting man. It may be up to 7 centimeters long and maybe more than a centimeter in width, and usually it is found in the intestine. So the smallest and the largest fluke are found, infecting man are found in the intestine, while you can find a couple of parasites that are living in the vascular system like schistosomes, in the lung like Fasciola species, and in the lung like Paragonimus westermani. So precisely, we can say that the trematodes of the fluke are divided into what is known as blood flukes, and we are going only to study today examples of those parasites, and also are going to deal with it briefly. So the flukes of the blood are Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematobium. I'm mentioning here only 3, but there is so many species, other species, but these are the important Schistosoma species causing infection in man. There is just at this stage, I would like to say there is what is known as lung flukes, which we have mentioned, for example, Paragonum zostermani, liver flukes, like we did not here mention, only mentioned Fasciola hepatica, but there is another species known as Fasciola gigantica. Intestinal flukes, we have Phaseolopsis bruschi and Heterophus. So our important parasites or our important trematodes or flukes are the blood flukes, which are found in the vascular system. So the disease bilharziasis or bilharzia or schistosomiasis is caused by 3 important species of schistosomes. In this diagram, for example, this is an experiment in the laboratory in the mice. So you can see that inside the blood vessels here, you could see the dark, long worm, waving worm. These are the adult schistosomes in the vascular system. To your right, you can see there is a huge granuloma. The white circle with the dark central part is an egg which is surrounded by what is known as granuloma. So precisely, the adult parasites usually are found either in the mesenteric veins, if they are Schistosoma mansoni or japonicum, or in the vesical veins around the urinary bladder if the parasite is Schistosoma hematobium, and they release eggs. The eggs usually are voided either in urine in hematobium or in the intestine of all other species like mansoni and japonicum. But some eggs during this process of moving from the mesenteric or vesical veins, they could be trapped and they can enter the circulation and may be carrying, for example, carried to the lung, to the liver. And usually they are lodged. Of course, how tight the eggshell may be, still there is a small pores through which some material or protein they ooze out or they come out from the egg, and these oozing small molecules are inducing an immune response which result into the formation of granulomas. So there is a recruitment of many cells like basophils, eosinophils, fibroblasts, etc. to come to form a circle or a resistant area around the egg to encirculate it from the immune response. Usually the disease here, of course, you can see there is change in the morphology and in the anatomy of the liver. And if there is so many granulomas, it can lead to fibrosis. Either it is an advanced fibrosis or almost partial fibrosis. And if there is so many granulomas, it can lead to liver dysfunction and other consequences. So the trematode of importance are the schistosomes, and here we are showing the adult parasite to your left, which is scanning electron micrograph. The outer side with a rough surface is the male, and it has at the anterior end a sucker, and down below it is another circle which is also a ventral sucker. The other one, the upper one, is the oral sucker or mouth through which the mouth opens, and the down one is known as the acetabulum or ventral sucker. So the function of the suckers is for attachment, and the male is almost elongated and flat, but it curves to form a groove known as gynecophoric groove through which the female is carried. Of course, the scanning electron wave to your left, but just stained parasite are to your right, showing both male and female. And down, you see an elongated structure with almost a divided tail. This is the infective stage of the parasite, which is usually found swimming in water. These stages, known as cercari, they are released from infected snails. In the center, for example, to your left, you will see the egg, and it has almost a lateral spine. This is the egg of Schistosoma monsoni. If you look to the one to the right, it has what is known as terminal spine. This is an egg of Schistosoma hematobium. The rounded egg down with no clear spine is Schistosoma japonicum egg. So precisely the three important species of schistosomes can easily be differentiated or diagnosed by the structure of their eggs. Lateral spine in case of Mansoni, terminal spine in case of hematobium, rounded with rudimentary small spine is the egg of Schistosoma japonicum. All schistosomes, in actuality, are transmitted or they need intermediate host in their life cycle. And usually, these snails are freshwater snails. What you see here are snails which have been collected from Oman. The upper snails, that is snail number A, B, and C, all of them, they belong to the same species known as Limnia. And Limnia snails are intermediate host for Fasciola species. But to the best of our knowledge, there is no problem with Fasciola among animals here in Oman. Fasciola basically is an animal parasite, but few human infections can happen. And the parasite will live in the liver of the animal or liver of man. When you come to the snails which are tiny and look somewhat fragile, number D to F, these are known as Bulinus riti. They have been collected here from a bottle body in Dhofar, and these are the intermediate host for Fasciola hepatica, causing urinary schistosomiasis. To the right, that is snail number G and H, which are rounded. These are bioinfilarious snails, and they are the intermediate host for Schistosoma mansoni. We don't have here the snail host for Schistosoma japonicum, but in the laboratory we have it, which I brought from China in one of my visits or conference visits. The snails which are down, and all of them look conical and sharp, known as Melanoides tuberculata. These are very common here in most of the freshwater springs or afflage in Oman. But to the best of our knowledge, these snails are of no medical importance. So please, if you see the bioinfilarious snail or the blind snail, be careful because just these are intermediate hosts for Schistosoma mansoni and Schistosoma japonicum. This is a water pond from the Green Mountain or near Salala, and this pond is a good swimming place for many teenagers. I have been there and have seen so many children every now and then swimming there. This has been identified as a site of transmission because from the school around this area, we got a couple of students or children who were infected with Schistosoma mansoni. The Minister of Health or the Public Health Affairs in Dhofar, they recognize there is a problem with mansoni, and they tried, for example, to put warning signs, warning people that this place could be transmitting Schistosoma mansoni. But as you see here, children, they don't mind, and they don't give attention to that, and they're just enjoying swimming and bathing in the water. This is from Dhofar as well. So the blood flukes, which are known as schistosomes, the important ones are Schistosoma mansoni and Schistosoma hematobium and japonicum. Schistosomiasis is almost infecting about 250 million people, but mainly in the tropical countries, and it is mainly among rural areas or among farmers who use irrigation canals, and where the irrigation canals are good environment or habitat for the snail intermediate host for schistosoma species. The parasite cause schistosomiasis or bilharziasis, and the name bilharziasis or bilharzia is after the German medical doctor who worked in Egypt, and he contributed to our knowledge of schistosomiasis. The adult worms, usually, as I said, they live in the blood vessels around the intestine, except in case of Schistosoma hematobium, which lives in the vessels around the urinary bladder, known as vesical vessels. How is it transmitted? An infected person will pass the eggs either in stool or in urine, in water. When the eggs reaches water, they hatch to release a swimming state known as menocydium. So menocydia are released from eggs, and they may penetrate any snail, but they only develop in the appropriate snail host. So in case of Mansoni, it is usually the bioinflaria snail, and in case of hematobium, it is the Bolinus snail. So different species of Bolinus snails and different species of bioinflaria snails are vectors of Mansoni and hematobium respectively. So the parasite will multiply in the snails, and they go through different processes of forming daughter sporocyst and mother sporocyst, and then from the daughter sporocyst they release infective stages known as the cercaria. And you have seen the cercaria as having an anterior part and a long tail, and the tail is divided, which is known as biforked tail. So infection is by the penetration of the cercariae, which are released in water. So they penetrate the skin, and usually it may take, the snail usually after penetration is lost or before penetration is lost, and the anterior part becomes almost elongated and somewhat oval. It has a rough surface, so it wriggles in the dermis, epidermis layer of the skin. And it may take it 1 or 2 or 3 days to go through it and then enter circulation. If that schistosomulum is not killed at the skin level, then it is standing a chance to live for a long period. It may live up to 30 years. So entering the circulation, it will be carried to the liver, and there they stay for a couple of weeks to develop to be adult parasite, and the male and female parasite they copulate, and then they migrate to the vesical veins or to the mesenteric veins, and they can live for 30 years. The females will release eggs, and the eggs have to be boiled to reach the lumen of the intestine and pass in a stool, or if it is a hematobium, most of the eggs will be passed out in urine. The pathology, the penetration of cercaria of the skin may cause dermatitis or some sort of allergic reactions, pruritus, and edema. Depends on the intensity of number of cercaria penetrating the skin. In Schistosoma japonicum infection, when the parasite become adult and start to lay eggs, so with the onset of egg laying, it causes a syndrome known as Katayama syndrome, which is, for example, enlargement on the lymphatic system, lymphadenopathy. It may cause malaise, and it starts to develop splenomegaly, causing abdominal pain, diarrhea, and maybe some blood in the stool. This Katayama syndrome can go for 1 or 2 months, and then it is automatically subsiding down. The eggs which are trapped in the tissue, especially in the liver, can cause granulomas. And as I said at the beginning, the granulomas can lead to advanced, that is partial or almost advanced fibrosis of the liver with liver dysfunction. So usually the disease here is an immunopathology because it is due to the granulomas. The granulomas will be changing the anatomy and the physiology and the structure of the liver because of the fibrotic formations. These are children. If you look to your right, there is the X in the middle, and you can see those large granulomas. The young boy who is standing, usually he's 16 years of age, but still he's showing retarded growth, massive ascites, advanced emaciation of the limbs, and it means this is an advanced stage of hepatosplenomegaly, ascites, and it may be a fatal situation. The young girl who is sitting, in actuality, it's not infected by Japonicum, but by another species similar to Japonicum, which is Schistosoma mekongii. However, mekongii is not given much attention because it is of limited distribution and prevalence. So such advanced cases could be fatal. In summary, Schistosoma mansoni and Japonicum can cause diarrhea, especially during the Katayama syndrome. That is the start of egg laying. They can lead to portal hypertension. They can lead to hepatomegaly, ascitic fluid, pseudotubercles, because there is almost a very thick biliary system, and it can lead to what is known as clay pipe stem fibrosis of the liver. Schistosoma hematobium can cause hematuria, that is blood in the urine, and it may be in the old Egyptian history. When a child, for example, a teenager, starts to show blood in urine, at that time they think that he is reaching puberty, while in actuality he is passing blood in urine because of the infection. So usually the parasite is affecting young children, maybe 8 to other teenagers, because they are active and usually they don't listen to the advice. Painful dysuria, and also may be associated with bladder carcinoma. How to diagnose schistosomiasis? Usually by seeing the X either in stool, if it is a hematobium, Japonicum, or mekongii, or in urine if it is the X which will be usually recovered in stool are mansoni, Japonicum, and mekongii, and in urine usually it is found. Usually it is the hematobium parasite. Sometimes, especially in chronic infections, there is a tendency of the X to be trapped in the tissues. So the patient may be showing symptoms of schistosomiasis, and then you examine the stool or urine and there is no X. But if you do, for example, rectal biopsy and bladder biopsies, you may see the egg there. There is various serological tests like ELISA, indirect hematogenation assay, and indirect fluorescence assay. The drug of choice is praziquantel, but there is other medicines. Control, of course, precisely I would say by treatment, by snail control and education and good sanitation. People should not pass their eggs, their stool in the open, which could be reaching water by rain or by animals kicking the stool, reaching water, and the eggs will hatch to infect the snails and the life cycle. Although I'm giving here only one line, that's control is treatment, snail control, education, and sanitation, but still you can give a whole lecture about how to control the snail. In principle, you can use chemicals against the snails. So the chemicals against the snails, they belong to a scientific group known as mollusk. So technically you use molluscicides. As against insect, you use insecticides. Against the mollusk, you use chemicals known as molluscicides. Also, you can use what is known as biological control. So you can introduce other animals like fishes that can eat the eggs and the baby snails. Also, you can use animals which are competitors. They don't eat the snail, but they can compete for the food, and they are more stronger and efficient, so they can starve the snail, and this is a biological control. Or it could be environmental. You make the Water, for example, not slow running water or stagnant water. You make it fast running water, and this is not suitable for the breeding of the snail. So, in principle, it is, let us say, chemical control, biological control, as well as education and sanitation. Thank you very much. I wish you all the best. And this brings me to the last lecture. I tried to make this large subject with so many difficult names uh, as precise and just kept to the very basic information. Good luck and see you next time. Bye bye.